Okay, hi everybody, this is Lilith, and I'm one of your TAs for BIBC 102. In this video, I'll do a review of lecture 11 and 12. Okay, let's begin. So, in the beginning of lecture 11, we talked about the coupling of ETC and ATP synthase. And we talked about what this mystery is right here. And we learned that this is the electrochemical gradient, right? Or the chemiosmotic hypothesis. So many years ago, scientists thought that ATP is produced by substrate level phosphorylation like we saw in glycolysis. But that's not the case for ATP synthase in the inner membrane of the mitochondrion. What happens is ADP is phosphorylated and turned into ATP by this huge enzyme called ATP synthase, which uses a gradient of protons and their fall back into the matrix through ATP synthase. And this energy allows for this subunit of ATP, well, the whole thing, to rotate, and that rotation changes different conformations in the F1 portion of the ATP synthase, and those conformations allow the phosphorylation of ADP. Okay, so ATP is formed by oxidative phosphorylation instead of substrate level phosphorylation. Okay, so how do we make this proton gradient? What is a gradient, right? A gradient is when we have a lot of something in one place and we have only a few of those things in another place. And so now these protons are looking for a way to come into the place where there's not a lot of protons. Okay, so that is the protons moving down their gradient. Okay, how is this gradient made? It is made by complex 1, complex 3, and complex 4 because they are proton pumps that take protons from the matrix and pump out into the intermembrane space. Okay, this constant pumping of protons allows for the concentration of protons to be very large in the intermembrane space and very small in the matrix. Okay, so here's a picture on the right that shows just that. Think of this proton pump as one of the complexes 1, 3, or 4, and see how they take the proton from the matrix and pump it into the intermembrane space. Now there's a lot of protons out here, and you see how the net charge out here is positive, and the net charge out here is negative, right? Or less positive. And also notice that there's simply a lot more protons out here and not a lot here. So this is what we mean by the electro, because we have a charge difference, right? Electrochemical, because we have a chemical difference. A lot of proton concentration here and not a lot here. So due to this electrochemical gradient, if there is a way into the matrix, those protons will come back in here. Okay? So, the ATP synthase is able to work and produce ATP because there is that gradient. So if there's no protons out here that can flow inside and generate that energy to make ATP, ATP will not be made, and ATP synthase will not be active. So you can tell how this gradient is responsible for ATP synthesis activity. And what makes the gradient? ETC does. So you see how ETC makes the gradient that allows ATP synthase to work. So to put it in a metaphor, right, it would be this. If there is no ETC, then there will be no proton gradient, and ATP synthase won't work, and we will not get any ATP production. So you see how all three of these processes are connected. One is dependent on the other, and that is what coupling between ETC and the ATP synthase actually means. So again, to reiterate, ATP will not be produced if ETC is not coupled to ATP synthase, right? And the opposite is also true. ETC makes the electrochemical gradient, which drives ATP production by allowing ATP synthase to work. Okay, now how do we actually know that these two are coupled? Well, researchers are able to measure oxygen consumption. And how do you use this oxygen consumption is you take molecules of succinate, you add it to an ETC. So we take succinate, and because succinate is the substrate of complex 2, then ETC will start to work and at the end produce water and reduce the amount of oxygen, right? Because it's used to make water. So that is how researchers measure oxygen consumption. And ATP synthesis, how we can measure that? We can synthetically make an electrochemical gradient and then see how much ATP is being produced. Okay? So because we can measure those two things, what we can do is first add ADP and phosphate. And when we only add ADP and phosphate, nothing is really happening, right? No oxygen is consumed and no ATP is synthesized. It is only when we add succinate, meaning we give something to the ETC to start working, that is only then when we start to consume oxygen and produce ATP. Okay? When we add cyanide, which is a molecule or a chemical that messes with the ETC, then you see how both oxygen consumption and ATP synthase are stopped. Now, when we add cyanide that messes with ETC, right, it makes sense how oxygen consumption is stopped. But we see that ATP production is also stopped, which confirms that these two are coupled. Okay, another experiment is, again, we're looking at oxygen consumption and ATP production. We first add succinate, so ETC has a substrate to keep working, right? But it doesn't work because we're not consuming any oxygen. See, this line is flat. However, once we add ADP and phosphate, when ATP synthase has substrates, right? ADP and phosphate, when it has substrates, oh, this looks like a face. Did you see that? Okay. When we add ADP and phosphate, then our oxygen consumption and ATP production both begins to rise. Okay? But when we add a molecule such as oligomycin, which messes with the ATP synthase, that is when both oxygen and ATP synthase stop. Again, both of them stop because ETC is coupled to ATP synthase. Okay, so how do we know, or how do researchers know that the electrochemical gradient is the actual driving force for ATP production? Well, when you want to know the role of something in a reaction, you can do two things. You can remove it and see what happens, or you can generate it and see what happens. So how do we remove the electrochemical gradient? Well, we can use weak acids, weak organic acids. I should probably add organic. Okay, and when we add weak organic acids, they help to dissipate the proton gradient, right? And if there is no more proton gradient, there will no longer be ATP synthase activity. And if there's no ATP synthase activity, and because it is coupled to ETC, right, then we will also stop oxygen consumption, okay? We will uncouple these mechanisms. Okay, and that is how we stop the proton gradient, by using weak organic acids. If anybody needs an explanation on the weak organic acids part, I'll go over it very quickly here zoomed in. Hopefully it's not too blurry. So when we have, let's say, let's do this. Let's say we have an acid, right? A negatively charged organic acid. When this is charged, then it will like to stay in the water compartment, right? Which is either the intermembrane space, cytosol, or matrix. However, if we give a proton to this negatively charged organic acid, then it will become protonated. And when it is protonated, it is found that it likes to, or it can, cross the hydrophobic membrane, meaning the protons are taken from the intermembrane space and they now can cross the inner membrane of the mitochondrion and go into the matrix. Okay, so you see how the point of this was to take a proton from the intermembrane space, cross it over the membrane, and finally have it end up in the matrix. So you see how this one proton, because it went through this process, it never went through ATP synthase. Okay, and every proton that doesn't go through ATP synthase, that is less and less energy that ATP synthase could use to form ATP. Okay, so that's how weak organic acids actually mess up the gradient. And to generate a synthetic electrochemical gradient, I believe the example Professor Hampton gave was to use bacteriorhodopsin. And bacteriorhodopsin is a membrane protein that, upon light, pumps protons into the matrix. Okay, and in that specific, I can draw it quickly. In that specific experiment, we had ATP synthase like this, in this, okay, and then we had bacteriorhodopsin, and when we shine light, then protons are pumped into here, and when there is finally a lot of protons in here, then they travel through ATP synthase and ATP synthase makes ATP. Okay, so this is how the proton gradient is generated synthetically through this bacteria rhodopsin. And then the scientists, all they did is measure how much ATP is being produced. Okay, so this further confirmed that this gradient is responsible for ATP production. Okay, so that's it for lecture 11. Now on to lecture 12. So before I move on, I wanted to talk about weak organic acids one more time, and also UCP, because I feel like this could be a foreign topic. So hopefully we can make sure that you all understand this. So this is our electron transport chain, and our protons are being pumped into here through these proteins, right? Hopefully all of you already have a good grasp of that. Then these protons get to flow through ATP synthase, give ATP synthase enough energy to rotate, right? And form, form ATP. Okay? However, if we have weak organic acids, then the acid plus proton will flow in here, and there will be less actual protons going through here. Okay, so that's what organic weak acids do. UCP is a natural uncoupling protein, and what it's a protein that spans the inner membrane, and it allows protons to flow through it. Now, when this happens, hopefully you understand by now, ATP synthase activity decreases because there's not a lot of protons going through ATP synthase. Okay, and when protons go through here, they still release a lot of energy, but that energy isn't put into ATP synthase to produce ATP. It is only generated as heat. Okay, so that is why this protein is also called thermogenin, because it generates heat. Okay, and I believe brown fat, which a lot of newborn babies have, brown fat is the type of fat content or fat cells, adipocytes, that are brown in color because they have so many mitochondria, and they especially, I believe, have a lot of UCP, and because they have a lot of UCP, they form a lot of heat, and that's good for babies to be able to generate their own heat through brown fat because they're vulnerable, and that's just through evolution. And then once you grow up, you start losing that brown fat. Okay, now let's move on to how do we get the NADH from the cytosol into the matrix so that it can go through ETC. And also, how do we regenerate NAD+. Okay, well, there's two ways: glycerol 3-phosphate shuttle and malate-aspartate shuttle. So let's talk about glycerol 3-phosphate shuttle first. Glycolysis takes place. We produce NADH. Then this NADH comes close to the inner membrane of the mitochondria. NADH is used up by this cytosolic glycerol 3-phosphate dehydrogenase. We regenerate NAD+, and those electrons go onto this carbonyl of dihydroxyacetone phosphate. This carbonyl is reduced into an alcohol, making this molecule glycerol 3-phosphate. Then what happens? This mitochondrial glycerol 3-phosphate dehydrogenase uses FAD to reoxidize this alcohol into this carbonyl. Then this FAD turns into FADH2, which passes its electrons onto coenzyme Q, then to complex 3, then to cytochrome C, then to complex 4, then to oxygen. Okay, so you see how in this process we've done two things. We've generated NAD+, that can now go back out into glycolysis, so that glycolysis doesn't stop. And we've also taken the electrons off of NADH and we've shunted it into ETC. The next shuttle is malate-aspartate shuttle. And this may look a little scary, but we'll go through it. I like to start from this top left corner. We have a molecule of oxaloacetate. Oxaloacetate is reduced right here on the carbonyl by NADH, so it turns into malate. See, this is an alcohol. Then malate goes through a malate transporter. See, now malate is here. It is oxidized at this alcohol position by NAD back into oxaloacetate. Then oxaloacetate goes through an aminotransferase reaction or transamination reaction and turns into the amino acid aspartate. Okay? And how does this work? Is we take an NH2 molecule and we do a Schiff base reaction onto this carbonyl, turning it into something like this, which is the amino acid aspartate. Where does this NH2 come from? It comes from the amino acid glutamate. So as glutamate takes its NH2 group and gives it to oxaloacetate to turn it into aspartate, glutamate itself becomes alpha-ketoglutarate because it's NH3 turned into a carbonyl. Now that we have aspartate crosses the inner membrane back into the intermembrane space. Now it's here, aspartate. Then aspartate goes through another amino transamination. In this case, it gives its NH3 group to alpha-ketoglutarate, which turns into glutamate. And because aspartate gave away its NH3, it turns back into oxaloacetate. Okay? Also notice that alpha-ketoglutarate from here can cross through the same transporter and be here, and then glutamate can cross through this transporter and be here. That is why this transporter is called malate alpha-ketoglutarate transporter, because it transports malate and alpha-ketoglutarate. And this one is named glutamate aspartate transporter because it transports aspartate and glutamate. I advise you to walk through this whole thing by yourself, because if you explain it to yourself and you say it a couple times, it'll definitely be ingrained in your brain better. So the whole point of this shuttle, again, is to take NADH from the intermembrane space and put it in the matrix so that it can go on to ETC. The other point of doing this shuttle is to regenerate NAD+, which is exactly what we did. Now this NAD+ can go into glycolysis. I did not spell that right. Glycolysis. Okay? And also notice that NAD+ from the matrix, right, ends up in here. And this is good because when NADH is used up by ETC, we produce NAD, right? Okay. Okay, another thing that I wrote here, cognate alpha-keto acid, I kind of went over it. So the cognate alpha-keto acid of the amino acid aspartate is oxaloacetate. If you look at both of these structures, the only thing different between them is this part. Okay? And then the cognate alpha-keto acid of glutamate is alpha-ketoglutarate, right? If we look at their structures, the only thing that's different are these. Okay. Another thing I want to mention before we move on is acceptor control. Acceptor control is when you have a substrate for something, then that substrate drives a reaction. For example, when there is ADP and phosphate abundant, then they drive ATP production. It's like ADP and phosphate say, hello ATP synthase, we're here, so start doing your thing so we produce ATP, okay? Now, if there is no ADP and phosphate, then ATP production will stop because they need these guys to make ATP, okay? And it is important for you to understand that this acceptor control actually regulates everything, even up to glycolysis. So let's go through that verbally because I have no space to write here. So if there is no ADP and phosphate, or ADP and phosphate are low, then ATP production will stop or will decrease, okay? ATP synthase activity will decrease. The electrochemical gradient will decrease. ETC activity will decrease. Not a lot of NADH and FADH2 will be used up in the ETC, right? So NADH and FADH2 will be accumulated, okay? That will back up Krebs cycle, okay? And then if Krebs cycle is down, then the accumulation of acetyl-CoA will make the glycolysis pathway backed up, okay? And so there will be an accumulation of glucose, okay? So you see how if there is a pathway, let's say A to B to C to D, and if we get rid of the last one, right, then everything before it will be stopped, okay? And in this case, A, B, C, and D are the pathways, right, or the processes. So like D can be ATP synthase, right? And then C can be ETC, then B is Krebs, then A is glycolysis, okay? Let's get rid of this art. Okay. So hopefully acceptor control makes sense. Now let's move on to gluconeogenesis, our first anabolic pathway that we're learning, okay? As you remember, anabolism is when we have smaller molecules and we're building bigger macromolecules out of them. Okay, so let's break down the word gluconeogenesis. Genesis of neoglucose. Neo means new. So we are making new glucose basically from scratch, right? So let's review glycolysis very quickly, and then we'll talk about the energetics of glycolysis, and then we'll talk about gluconeogenesis. So in glycolysis, we take glucose and we break it down all the way till pyruvate. Okay, and as you remember, there are 3 steps that are big energy drops. Step 1, Step 1, Step 3, okay, and the last step. And here is the delta G drops of glycolysis. Step 1, Step 3, and Step 10, I believe, the last step. So because these are huge negative delta Gs, the whole glycolysis pathway is exergonic, meaning this happens spontaneously. Imagine if we say, well, now let's go back, right? This will not happen spontaneously. This is very non-spontaneous, because imagine we have to climb this big energy ladders. Oh my God, what was that? These big energy ladders, right? And that's just not energetically favorable, so it will be very endergonic. So what the cell does is it has found a clever way of bypassing these reactions where there is a big energy drop. And those bypass reactions are at step 10, step 3, and step 1. Okay? So, from pyruvate, we use ATP, pyruvate carboxylase, right? And remember what carboxylases do? They carboxylate molecules. They use bicarbonate, right, and biotin. And we take pyruvate and we put an extra carbonyl on it, or a carboxyl group, and we form oxaloacetate. Okay, then we use a molecule of G-3P, the enzyme phosphoenol pyruvate carboxykinase, or I like to call it PepsiK. Okay, and that turns oxaloacetate into phosphoenol pyruvate. Now, we're done with the first step. That takes a lot of energy. The rest, all the way up to here, is easy peasy, right? Same reactions going backwards. Okay, then we get to over here, and this is step 3, which is also another big drop of energy. So we need another bypass reaction. And this bypass reaction takes fructose 1,6-bisphosphate and cleaves off the phosphate that's on the first position. How does it cleave? It cleaves with a phosphatase enzyme. A phosphatase enzyme is a hydrolase enzyme that takes water and plucks off one molecule of inorganic phosphate. You don't actually need to know that hydrolase part. If you're going to take the MCAT, you want to know that, but not for this class. But it's just easy to remember because phosphatases use water to take off a phosphate group. Okay? Then the last bypass reaction is the first one. Same idea. We have a glucose 6-phosphatase that uses water and takes off this 6th phosphate. And there you go. We formed glucose all the way from pyruvate. So that's gluconeogenesis. And I've added a closer view of those bypass reactions over here, so you're welcome to look at this in the PDF notes or here in the video, but because I went over it here, I will skip over that. Okay, now, one enzyme or one reaction that we really need to talk about because it's very important, it is the reaction that takes us from fructose 6-phosphate to fructose 1,6-bisphosphate, or from fructose 1,6-bisphosphate to fructose 6-phosphate. Okay, so let's look at this reaction first. It is done by the enzyme PFK, okay, phosphofructokinase 1. And you remember this from when you studied glycolysis, hopefully, okay? And if you remember, this enzyme is activated by something called fructose 2,6-bisphosphate. And at the time, before the midterm, we told you, we'll talk about that later. Just remember that it activates this enzyme for now. And so now is when we talk about it, okay? The molecule fructose 2,6-bisphosphate activates phosphofructokinase 1, and it inhibits fructose 1,6-bisphosphate, okay? Now let's take a closer look on how this happens and why this happens. To understand this better, let's look at the structure of fructose 2,6-bisphosphate. We have our fructose 2,6-bisphosphate. So carbon 1 is here, 2, 3, 4, 5, 6, 2-phosphate, and 6-phosphate, okay? How is fructose 2,6-bisphosphate made? Well, it is made from fructose 6-phosphate by the enzyme phosphofructokinase 2. Okay? Now, make sure to understand that phosphofructokinase 1 makes fructose 1,6-bisphosphate, but phosphofructokinase 2 makes fructose 2,6-bisphosphate, okay? Then, to take fructose 2,6-bisphosphate and turn it back into fructose 6-phosphate, we have this enzyme, fructose 2,6-bisphosphatase. Sorry, that took me a while. Yes, fructose 2,6-bisphosphatase 2. So you see how on this left side we have fructose 1,6-bisphosphatase. I'm guessing 1. And this is the phosphatase that takes off an inorganic phosphate off of this. Okay? So you see how there are many parallels. So this can get difficult because you might confuse all of these enzymes, but make sure to go over them, pause this video, and make sure you understand the difference between them. Okay, now why is it important? Why do we need to know this? Let me tell you. Where does fructose 6-phosphate come from? Let's look in glycolysis. Where does fructose 6-phosphate come from? Well, it comes from glucose 6-phosphate. Where does glucose 6-phosphate come from? It comes from glucose. So this means that if there is fructose 2,6-bisphosphate present, then we know for sure that there is glucose present. And vice versa. If there is glucose present, then you better know that fructose 2,6-bisphosphate is going to be made, because fructose 2,6-bisphosphate is simply made from glucose. Okay? This is important to remember because this is one piece of information that will help you a lot when there are questions about the interplay between all of these enzymes and their substrates. Okay. So, let's talk more about how fructose 2,6-bisphosphate activates phosphofructokinase 1 and inhibits fructose bisphosphatase 1. One second, let me check something. Okay, sorry, I had to check something in my mind. Okay, so when there is fructose 2,6-bisphosphate, right, I just told you that if this is present, then we should keep in mind that there is glucose in the cell. Okay, so let's think about this critically. If there is a lot of glucose in the cell, what would the cell rather do? Would it want to break down this glucose to make ATP and to live and to function? Or would it want to ignore these high levels of glucose and make even more glucose through gluconeogenesis? The logical answer here would be that it would like to break down this glucose, right? Because it has food, it has glucose, it wants to break it down to form ATP. So logically speaking, which helps most of the time, right? When there is fructose 2,6-bisphosphate, it means there is glucose present. This means that glycolysis is going to be favored because we want to break down that high levels of glucose. So you see how the presence of F26BP activates the enzyme that allows glycolysis to go on. Okay? And at the same time, if there is fructose 2,6-bisphosphate, because there is glucose, the cell wouldn't want to make any more glucose through gluconeogenesis. So it will inhibit the enzyme that allows the cell to do gluconeogenesis. Okay, so you see how because we understand that this comes from glucose, and because we use logic to see what would the cell want to do with the glucose, we can understand how these processes work and why this is activated and this is inhibited. Okay, so please practice this. Let's go over these graphs because this might not be very intuition friendly for some people. So remember that, wait, no, don't remember anything. Fructose, right now in this graph, we're looking at phosphofructokinase 1 activity, okay? And you see how when there is no fructose 2,6-bisphosphate, the activity of PFK1 is very bad, right? So it is not very active when there is no fructose 2,6-bisphosphate because there's nothing telling it to activate. But when we add fructose 2,6-bisphosphate, it activates PFK1. And see, the activity of PFK1 is activated. Let's look over here. Fructose bisphosphatase 1. When there is fructose 2,6-bisphosphate, it inhibits the activity of fructose bisphosphatase, right? We don't want to do gluconeogenesis, so we are going to inhibit this enzyme. But when there is no fructose 2,6-bisphosphate, because there is no or low glucose, then you see how the activity of fructose bisphosphatase 1, which is in gluconeogenesis, is activated. Okay, so hopefully you understand that when there is no fructose 2,6-bisphosphate, there's no glucose, so gluconeogenesis is favored. And when there is fructose 2,6-bisphosphate, then that means there is glucose and glycolysis is favored instead. Okay, and let's talk about how exactly these two enzymes work in terms of their activation and inhibition. So when PFK2 and fructose bisphosphatase 2 are not phosphorylated, then this state of this enzyme favors PFK2 activity. Okay, so this is something you just have to memorize, right? Not phosphorylated, PFK2 is active. Okay, when it is phosphorylated, then fructose bisphosphatase 2 is active. Okay, so let's see what let's see what glucagon and insulin do. So glucagon is released from, I don't know if you learned this, but alpha cells of the pancreas, right? Pancreatic cells. And when there is glucagon is secreted when there is low glucose, right? Professor Hampton likes to say glucagon is present when glucose is gone, right? Gone. I think it's funny. I definitely remember it like that. So when glucose is gone, glucagon is secreted. So again, think logically. Low glucose, what would the cell want to do? It would want to make glucose, right? And which process makes glucose? Gluconeogenesis. So you already know in the back of your mind that glucagon is going to activate gluconeogenesis. And that's exactly what it does. It activates a kinase protein that takes ATP and phosphorylates this thing. And as we said earlier, when it is phosphorylated, then it favors the active form of fructose bisphosphatase 2. And as we know, what does fructose bisphosphatase 2 do? It takes fructose 2,6-bisphosphate, right? And it breaks it down into fructose 6-phosphate. So it will degrade the amounts of fructose 2,6-bisphosphate. And if there is less fructose 2,6-bisphosphate, then glycolysis will not be stimulated, and instead gluconeogenesis will be stimulated. So again, glucagon favors gluconeogenesis. Now let's go through what insulin does. Insulin is secreted from the beta cells of pancreatic islet cells. I don't remember if Professor Hampton mentioned that, so if he didn't, don't know it, but if he did, it's good to know. So insulin is secreted when glucose levels are high in the cell. Okay, and let's think logically again. If there's a lot of glucose, what does the cell want to do? It wants to break down that glucose. So insulin will favor glycolysis, and that's exactly what it does. Insulin activates an enzyme, phosphatase, that takes water and hydrolyzes this phosphate off, and now this enzyme is unphosphorylated. And as we said earlier, the unphosphorylated version favors the active form of phosphofructokinase 2. Okay? And what does phosphofructokinase 2 do? It takes fructose 6-phosphate and phosphorylates it at the position of the second carbon, forming fructose 2,6-bisphosphate. So now that the amounts of fructose 2,6-bisphosphate are increased, they will stimulate glycolysis and in turn inhibit gluconeogenesis. So you see how production of insulin eventually stimulated glycolysis. So again, do practice this because it is good if you go over it a couple of times yourself. And it seems like that this is the end of what I had for lecture 11 and 12. Sorry, I feel like this video was very long. If you have any further questions or clarifications or any comments, please don't hesitate to email me or message me on Canvas. My name is Lilith, or you can visit my office hours on Mondays at 7 o'clock. I was never able to place the password anywhere. Oh, maybe I can do it in my, I will post the password to my office hours in my Canvas IATA page, okay? But if somehow I forget, please email me for it. Okay, thank you and good luck.